The overall goal of this procedure is to effectively deliver solutions into the cytoplasm of livestock zygotes using a blunt-end needle with the aid of a laser.
This method is used in research and in the animal transgenesis field, since when associated with a genome editing tool such as the CRISPR-Cas9 system, it can create knockouts to study gene function and transgenic animals in a very simple and reliable way. The main advantage of this technique is that it can be used to deliver any solution into the cytoplasm of large animal zygotes with 100% injection efficiency, minimal lysis, and normal developmental rates.
First, prepare the injection micropipette by placing a borosilicate glass capillary into a micropipette puller and locking it into place. Use an appropriate program to pull the capillary so it results in a thin tip with a long taper. Then carefully remove the pulled pipettes from the device and place them onto a microforge in a horizontal position. Bring the pulled pipette and the microforge heater filament with its glass bead into focus. Use the eyepiece reticle to measure the desired thickness and move the pipette horizontally until the heater filament reaches the target inner diameter of 5 micrometers. Adjust the heat to about 45% and gently bring the pipette down so it touches the filament. Then, briefly activate the heater. This will slightly melt the pipette so it adheres to the filament, and upon cooling, the pipette will break at the contact point, generating a straight cut.
Setting the right temperature is key in this step. Temperatures too high will bend the pipette, while temperatures too low will not be sufficient for cutting the pipette.